When you think about the nervous system, you want to think about dividing the nervous system into three separate functions: input, processing, and output. If you can think about these three different processes as we go through the information, that can be helpful in organizing the functions of the nervous system. Think about a computer. It has an input. It's like the keyboard and the mouse. That's how it senses what's going on in the world around it, one of the many ways. And then processing occurs in a black box, and then the computer responds with the screen, the printer, other ways as well. I make this point because just because you turn the computer screen off doesn't mean the processing or input has stopped. If you disconnect the keyboard, processing and output still work. And this is interesting because we see this in the nervous system as well. Alright, so this is my picture of a neuron. The neuron is the basic cell of the nervous system. And input is received at the cell body. It receives neurotransmitters from other neurons. And then once that cell body has processed the information, it sends its output down an axon, a long tube, and that long tube carries an electrical signal called an action potential. So the electrical signal carried down the axon is called an action potential, and that's why I show that lightning bolt going down the axon. Something to keep in mind is an action potential only goes one direction, in this case from the cell body to the end. And so neurons have electrical signals that go down their axons, and those signals only go in one direction. Something to keep in mind is this concept of myelin. Myelin insulates the axon for fast action potential. Don't want to talk about nodes of Ranvier and saltatory conduction today, but I did want you to know that myelin provides this insulation for the axon, and that makes the axon's action potential run more smoothly and more quickly. And myelin is made of glia cells, and they're like little flat pancakes that have wrapped around a tube, and we have a picture of that. And glia comes from the word glue. So here we've sliced through an axon. We're looking at the center of the axon, and we can see this myelin is wrapped around the axon, providing the axon electrical insulation so the action potential can go faster and more efficiently. Which brings us to a disease called multiple sclerosis. Multiple sclerosis is a demyelinating disease that results in weakness and other problems as well. Because healthy myelin is essential for nerve conduction and action potential, diseases like multiple sclerosis that destroy myelin can result in all sorts of neurologic problems, including weakness. So the action potential going down the neuron causes a neurotransmitter to be released from the neuron, and that's the little red dots coming from the end of the neuron at the synaptic terminal. And that's supposed to show neurotransmitter being released by the neuron in response to action potential. So neurotransmitters are chemicals released from neurons to communicate with muscle. Neurons communicate with muscle. That's how your muscles move. Neurons signal them. Neurotransmitters can come from the neuron and signal organs like heart rate going up, heart rate going down. That's due to neurons that secrete neurotransmitters on the heart's conduction system. And then, of course, neurons can communicate with neurons, and billions of them do. So some neurotransmitters that you guys will talk about during this section are acetylcholine or acetylcholine, depending on who you argue with. The catecholamines are dopamine, norepinephrine, and epinephrine. They are a family of neurotransmitters that are made from the amino acid tyrosine. Serotonin, 5-hydroxytryptamine, that comes from the amino acid tryptophan, and gamma aminobutyric acid, that's GABA. GABA is just fine if you can't say gamma aminobutyric acid, and I'm sure you don't know too many people who can even say that. Try to say gamma-aminobutyric acid 3 times fast and tell me how you do. Well, somebody once asked me, does a neuron just secrete all of these neurotransmitters? And I thought, well, that would be confusing. And so in this course, one neuron secretes one neurotransmitter. And so each type of neuron is going to secrete one type of neurotransmitter, like acetylcholine, which is used in the neuromuscular junction, meaning the neuron that signals the muscle only uses one kind of neurotransmitter, and that's acetylcholine. And norepinephrine is going to be used by the sympathetic neurons at the end organ. So the sympathetic neuron that innervates the heart and speeds the heart rate up, that's norepinephrine. At least in this course, you might read that teeny tiny amounts of epinephrine are involved, and that's because the catecholamines are family. But for this course, one neuron, one neurotransmitter, and you'll be doing just fine. Alright, so a synapse is that extracellular space where these neurotransmitters are released from one neuron onto either the muscle, the organ, or another neuron. And so let's take a close view at where this neurotransmitter is released from the synaptic terminal. And I have that circled in red, and I'm going to try to animate that for you. And so here is that synaptic terminal of the neuron. We've blown it up and redrawn it for you. And here the little red dots are neurotransmitter, and they're stored in little packets that I have circled in blue. These packets that store neurotransmitter are called vesicles. So the neuron's flashing because action potential has come down the axon, and action potential causes the neurotransmitter to be released. And so action potential causes the vesicles to release neurotransmitter during action potential. I think we've belabored that point. So the neurotransmitter is released into a space called a synapse. And so this synapse can occur between a neuron and a muscle, a neuron and an organ, or a neuron and another neuron. And so you'll read about synaptic this and synaptic that. The synapse is that space where this neurotransmitter is released into. Which gets us to these terms presynapse and postsynapse. You can see the presynaptic membrane is that which is releasing the neurotransmitter, and the postsynaptic membrane is going to have some receptors that sense the neurotransmitter. And the words presynaptic and postsynaptic are used all the time in pharmacology and neuroscience, and that's just what those terms refer to. So on the postsynaptic side of the membrane are receptors that sense the neurotransmitter, and they look like little white teeth down on, at least on this cartoon. And so receptors on the muscle, receptors on the organ, receptors on a neuron are going to sense this neurotransmitter and pass the signal from the neuron onto whatever is receiving that signal. And so here I'm at the postsynaptic receptor. Here I have a neurotransmitter activating a sodium channel. And this is called a ligand-gated ion channel. Ligand just means any chemical like a neurotransmitter. Gated means that neurotransmitter opens the gate up. Sodium is an ion because it's positively charged, and the channel allows the sodium to rush back into the cell. So gated ion channels, like sodium channels, opening those ion channels causes a voltage change across the cell membrane, and this allows for instant on and off communication between the neuron and, let's say, the muscle. See, the neuromuscular junction uses acetylcholine from the neuron, and that activates receptors on the muscle, and that opens sodium channels. And so opening those sodium channels causes an instant voltage change on the membrane, so the muscle can contract. When the acetylcholine's gone, the sodium channel's shut, and the signal is off. And so you might hear this called ionotropic receptors or ionotrophic receptors, and these receptors open ion channels and allow the neuron to have instantaneous on and off communication with the postsynaptic membrane. which leads us to myasthenia gravis. It's a weakness caused by circulating antibodies that block the acetylcholine receptors at the neuromuscular junction, and this is going to inhibit the effect of acetylcholine on the muscle. Antibodies are immune globulins. They're made by the immune system, and they're designed to attack virus and attack bacteria and other foreign substances. But in some situations, your immune system gets confused and starts making antibodies against your own body parts. And in myasthenia gravis, circulating antibodies will block the acetylcholine receptors, preventing the neuromuscular junction from working properly. And this is the cause of weakness in myasthenia gravis. So, in myasthenia gravis, these antibodies are going to block the receptors in the neuromuscular junction. And here, on this animation, we see these little Y-shaped proteins. Those are the immunoglobulins blocking the receptors that should be sensing acetylcholine. Let's talk a little bit about second messenger activation. You might read about that. Second messenger activation allows complex chemical reactions to occur. In the neuromuscular junction, we opened sodium channels, which allowed a quick voltage change across the membrane, instant on and off. But when we want to speed the heart rate up, that's not an instant on and off thing. That's a gradual metabolic change that has to go on inside of the heart, and this is done with second messenger activation. This allows for metabolic changes. And you'll see this when a neurotransmitter controls organs like heart and smooth muscle. You'll see second messenger systems used like cyclic AMP. Cyclic AMP is a second messenger. So here's a neurotransmitter activating a receptor on the postsynaptic membrane, and that activates adenylyl cyclase. And adenylyl cyclase converts ATP, which is freely available as an energy source, but it converts this ATP into cyclic AMP, and cyclic AMP is our second messenger. And so here our neurotransmitter, rather than opening a sodium channel and causing a voltage change across the membrane, our neurotransmitter is raising cyclic AMP levels. And by raising cyclic AMP levels, cyclic AMP is going to interact with all sorts of regulatory proteins inside of the cell and cause metabolic changes to occur. And so you might hear these called metabotropic or metabotrophic receptors. And I just wanted you to see the difference between second messenger system activation and ion channel activation. Do not confuse nerve with neuron. All right, a nerve is a collection of neuron axon fibers, and the signals in nerves can run both ways. Even though the axons within them are one-way streets, nerves can be a collection of one-way streets, either all running in one direction, all of them running in the opposite direction, or all of them running in either direction. So a nerve is just quite simply a collection of axon fibers. And so if you took a nerve and sliced through it and looked at it under the microscope, you would see these little neuron axon fibers in the center of it. It wouldn't be lit up like a Christmas tree, but it would look something just like this. Do not confuse nerve with neuron. A nerve is just a collection of neuron axon fibers.